Hello everyone, welcome back to the channel. If you are new here, my name's Zach Hancock. I'm an evolutionary biologist who specializes in population genetics, phylogenetics, and genome evolution. On this channel, you'll find a mix of evolution education videos like the one we're fixing to do, as well as sort of the history and philosophy of science and a bit of pseudoscience debunking, mostly focused on creationism. This is the third part of my ongoing series, The Causes of Evolution, where we are attempting to sort of dig into the first principles of evolutionary theory and really evaluate this idea at a quantitative level. And so just as to sort of recap the past two episodes, we started off with what is phenotypic variation, where does it come from, and what sort of variation is relevant to evolution. So we talked about how phenotypic variation can emerge from development, can emerge from the environment, or it can emerge from underlying genetic differences between individuals. And we concluded that what we're ultimately interested in an evolutionary context is the genetic contribution of individuals to succeeding generations. In the next video, we derived a very simple model based on just Mendelian inheritance called Hardy-Weinberg equilibrium, and we found that it implies that there should be no evolution, that allele frequency should not change from generation to generation under a series of sort of simplifying assumptions. And this told us something very important, that the processes of evolution actually lie in the various ways you can violate the assumptions of Hardy-Weinberg. So those are the mechanisms of evolution, and the first one that we talked about is the violation of random mating. So non-random mating, we found, was one of the first mechanisms of evolution capable of changing allele frequencies and genotype frequencies, but we found that many types of non-random mating change genotype but not allele frequencies, such as inbreeding and positive assortative mating. We found that negative assortative mating will change frequencies, but only for a little bit, that they will eventually equilibrate at 50% frequency and then they won't change anymore. And we also introduced the concept of FST, which measures the amount of genetic differentiation between populations and is hence sort of a direct measure of the amount of evolution that has occurred. So that's what we talked about so far. Let's now refresh our memories about what all of the assumptions of Hardy-Weinberg equilibrium are. So again, if these assumptions are met, then evolution is not expected to occur. Allele frequencies will not change across generations. One, that gametes are paired at random. We found last time that if you violate this assumption, then you will deviate from Hardy-Weinberg equilibrium. Secondly, that gametes aren't preferentially chosen from the gene pool, right? So that whichever gamete you draw at any one time is completely blind to which one it is. You have no idea which one you're picking, okay? Third is that allele frequencies are equal between the sexes. Fourth, that alleles don't change from individual to the gene pool. So whatever alleles that individual has are the only alleles that they can then give to the gene pool. They don't change in that process. Fifth is that gametes are drawn infinitely and with replacement. So we're drawing every possible combination that could exist in the gene pool. That's the fifth assumption. The sixth is that gametes aren't added from other gene pools. You can think about this as the gene pool is kind of a closed system. We don't have multiple gene pools in which individuals are being added from other gene pools, right? It's a closed system. And then finally, individuals only contribute to the gene pool once. We call this non-overlapping generations, where individuals aren't continuously adding their alleles into the gene pool across multiple generations. Okay, so again, these are the assumptions of Hardy-Weinberg, and if they are met, then we do not expect allele frequencies to change across generations, and hence we don't expect evolution to happen. What we've talked about thus far is non-random mating. That was the previous video. But today we're going to look at the violation of the second assumption, that gametes aren't preferentially chosen from the gene pool. And we should already kind of recognize what mechanism of evolution this one is. And if you guess natural selection, you are correct. So natural selection has the longest history of all the different mechanisms of evolution. because it is the original mechanism that was first proposed by Darwin in 1859 in The Origin of Species. And I want to stress here that his perception of natural selection is a bit different than the way that we think about it today. So what I want to do is give his perception as sort of a historical basis of where we're coming from, but then we're going to pretty rapidly diverge from the way he thought about natural selection to the way that the founders of, theoretical population genetics think about evolution. So first, Darwin put forward his argument for evolution by natural selection as a kind of verbal argument that goes like this. One is that individuals have far more offspring than can survive to reproduce. We call this reproductive excess. Secondly, is that there exists variation between individuals, and some of that variation is heritable. That is, as we talked about before, it has a genetic basis. Three, that some of that heritable variation provides an advantage to the individual, increasing the chance that they reproduce over others. Again, remember, not every possible offspring can reproduce. There is an excess reproduction. Only some of them will get to. So if they have some variation that gives them a little bit of an advantage over everyone else, then that particular trait, the fourth claim, will spread through the population. Okay, and he calls this process of differential reproductive success natural selection, and that spread of that new variation, evolution by natural selection. And framing it this way is very important, and that leads us to this next point, that natural selection is not the same thing as the theory of evolution by natural selection. R.A. Fisher, one of the founders of theoretical population genetics, famously began his 1930 book, The Genetical Theory of Natural Selection, with the sentence, "Natural selection is not evolution." And Landy and Arnold, I think, really summarize this well in their 1983 paper when they write, quote, "Natural selection acts on phenotypes regardless of their genetic basis and produces immediate phenotypic effects within a generation that can be measured without recourse to principles of heredity or evolution. In contrast, evolutionary response to selection, the genetic change that occurs from one generation to the next, does depend on genetic variation." So to kind of get a grasp on what this means and why we differentiate natural selection and evolution by natural selection, let's start with a simple conceptual model. So imagine just a haploid population that reproduces by cloning, okay? And you've got three different variants, red, blue, and green. The red variant, on average, only leaves half of an offspring every single generation, okay? So that means on average, half of them don't reproduce. The blue one, on average, only replaces themselves. So they only undergo fission one time, and so they leave one offspring on average. The green one will, on average, split twice, leaving two offspring. So with this understanding, and assuming that each one of the variants in these haploid lineages are what's contributing to their ability to leave more offspring, we can then take a couple of measurements. One, we can think about the average number of offspring left in the population. We can measure this by 2 plus 1 plus 0.5. Those are the three possible numbers that each one of the variants leaves. Divide them by 3, and that gives us an average of 1.16 offspring left every generation. We can then think about how we can conceptualize this at an individual basis by introducing the concept of fitness. And so the green's fitness, for example, we could define as 2, the number of offspring they leave on average, divided by the population average equals 1.72. So we can see that the number of offspring that green leaves on average is greater than the population average. Alternatively, blue on average leaves 0.86 and red 0.43, both below the population average. Another way we could do this is by relative fitness. So we could standardize the concept of individual fitness by the maximum. So if we take green, since green is the max, we can standardize by 2. So green's fitness is 2 divided by 2, which is 1. So they have the highest fitness. Blue's fitness is 1 divided by 2, so that's 0.5, so they have half the fitness of green, and then red's fitness is 0.5 divided by 2, which is 0.25, that is, they have only a quarter of the fitness of green. Okay, so with that in mind, we can now think about how fitness relates to natural selection. So differential reproductive success, as Landy was just talking about before, is natural selection, right? That's natural selection happening in the here and now. Green leaves more offspring than blue and red. That's differential reproductive success, hence that's just natural selection. Now, evolution by natural selection is this process actually changing the frequency of these variants through time. So here on the bottom panel, right here, I'm just showing kind of a made-up graph where green, you can imagine, increases in frequency at the expense of blue and red. So this change in frequency through time, driven by differential reproductive success that is connected, right, to the variance in genotypes, that is evolution by natural selection. Just wanted to make that clear, divide those two things up. Okay, so that brings us now to this thing that we kind of introduced without really talking a whole lot about, and that is fitness. So fitness is defined in many different ways, but they are all meant to capture an individual's contribution to the next generation. Fitness becomes complicated because, again, selection can act at many different states. So this little circle is just kind of showing the life history of a simple dioecious diploid species. You can see that from the zygote to the adult, this particular arrow can be acted upon by viability selection. So how likely are you to grow from a zygote into an adult and be able to reproduce? If everybody has exactly the same probability of doing that, then there's no viability selection, right? And so if we measured fitness right there, everybody has the same fitness. However, if we measured fitness from the mate choice, from being able to find a mate, then we would be talking about sexual selection. And then we can measure fitness as how likely are you to get a mate versus some other individual. So fitness can be measured there. However, if everybody has the exact same probability of mating, then there's no selection acting there. Then from the parent to the production of the gametes, there's another area where fitness could be defined. It could be defined by fecundity or by gametes, right? You can actually have selection acting on gametes themselves. And then lastly, from the gametes pairing up to the zygote, you can have compatibility selection. So what is the compatibility of the two gametes that are going to form that zygote? That's another area that selection can be acting. So again, people often complain about fitness as this, like super complex concept, but the reason for this is based on when you measure fitness. Hence, that tells us something about when selection should be acting, right? So just keep in mind that fitness is a multifaceted thing that encompasses each of these different life history components, all of which are capturing an individual's ability to contribute to the next generation, right? That's the key thing that connects all of these different types of fitness, is that they all can be boiled down to, is the individual going to contribute to the next generation? Irrespective of if it's viability, sexual selection, fecundity, compatibility, etc. That's the common thread across all. So then we can take a step back and say that the most exact and sort of general definition of fitness is at the individual level. So individual fitness, which can be expressed by the genotype level as well, because genotypes code for phenotypes. And that is just the number of offspring they produce, often divided by the population mean number of offspring. And this is a very useful definition of fitness because it permits us the ability to link fitness to ecology. Okay, because then it becomes a function of an individual's contribution to population growth. So with that in mind, let's see how measures of fitness in an evolutionary context are actually connected to the ability of the population to grow in size. So fitness can be measured as either absolute or relative, as we talked about before. Absolute fitness is the best way to connect it to ecological principles because it's going to be connected to population size. So absolute fitness is just the expected number of offspring produced by a parent of a given genotype. Previous example, the green lineage always left 2 offspring. So the expected number of offspring that they produce is 2. That's their absolute fitness. Again, their relative fitness is taking their absolute and dividing it by the highest absolute. Since they were the highest, their relative fitness is 1. By relativizing fitness, we then kind of divorce it from ecology, we divorce it from population size. But what we want to do here, since we want to really try to make fitness a concrete idea, is we're going to stick with absolute fitness and we're going to show how it's connected to ecological principles. So let's define the population size in the next generation as nt+1 is equal to p squared, this is the frequency of the homozygous genotype, multiplied by the population size in the present generation, n sub t, multiplied by the absolute fitness of that genotype. Okay, and we're doing that for each of the alternate genotypes, the heterozygotes in the middle, and then the alternate homozygote at the end. We should recognize that p squared plus 2p1 minus p plus 1 minus p squared is Hardy-Weinberg, right? That's the random mating expectation that we derived in the last video. All we are doing is adding in population size to each one of these and then multiplying that by the absolute fitness of each one of those genotypes, okay? Furthermore, we can define mean population fitness, which is W bar, as equal to the Hardy-Weinberg frequencies, the p squared plus 2p1 minus p plus 1 minus p squared, just multiplied by the absolute fitnesses of each one. Okay, so that's this expression here. Therefore, we can directly relate mean population fitness to the population growth rate. And in fact, it scales at a rate of the mean population fitness multiplied by the present population size. Doing that is going to give us the population size in the next generation. So I've just kind of plotted an example of this here. This is the mean population fitness with the population growth rate. So you can see as the mean population fitness increases, the population grows in size. So, okay, so this is really cool. This, again, allows us to connect fitness to something real, but we can also show that fitness in this context is connected to ecological principles of population dynamics. So if you are familiar at all with ecology, you might know about the population growth rate, which is often just called R, is a central parameter in just general population biology. For example, in the famous Volterra predator-prey models, R kind of plays a central role here. So in case you're not familiar with that, down here at the bottom is sort of like a seminal paper in which they're tracking the population size of rabbits, or hares in this case, and lynxes, their predators, right? So the hares are in the sort of dark brown, the lynxes are in the red. So through time, they're just tracking the changes in the population size. You can see that as the predator's population goes down, the prey's population goes up. As the prey's population increases, now there are more prey for the predators to eat, so their population goes up as well. But as their population increases, they begin to deplete the rabbits, whose population goes down, right? And so this creates this kind of oscillating effect through time. And the slope of that increase in size, the ability of that increase to occur, that's a central parameter in the Volterra model that is just called R, the population growth rate. So again, we can see that there's a real world interpretation of this value. So let's see if we can connect this directly to our concept of mean population fitness that's defined from a genotypic level. And sure enough, we can. So if we take the population size at t+1, so in the next generation, again, as we saw before, that's equal to n sub t w bar, which is approximately equal to n sub t e raised to the r, again, where r is the population growth rate. We can see that just rearranging these, the n's actually cancel out, and what we're left is that the population growth rate, the per capita rate from ecology, is approximately equal to the natural log of the mean population fitness. So to put some numbers in there, imagine a population of size 1,000 and a growth rate of 0.1. We can directly calculate mean population fitness from those with this expression as follows, just plugging in 0.1 to E and we get 1.1. That's the mean population fitness. Just going backwards, plugging in the natural log of 1.1 gives us 0.095, which is approximately 0.1, which was the population growth rate that we started with. So again, I just really want to stress this because some critics of natural selection, and you know who you are, often claim that fitness is not a real thing. It's not something that has any like real world meaning to it. And I just wanted to kind of go through this to show you that mean population fitness has a very, very real world application with the concept of population growth, and that we can actually get the mean population fitness directly from measuring the rate of growth of a population. Okay, so now that we've been able to connect these two things, right? We've been able to connect the mean population fitness to something we care about in ecology. That then means this mean population fitness has a lot of things that we could be interested in. And one of those is, how does the mean population fitness change with allele frequencies? Okay, so as the allele frequencies are changing, does the mean fitness also change? Right, so to be able to sort of get at this question, what we need to do is actually take the derivative of the mean population fitness. So to put this in kind of plain English, the rate of increase of mean population fitness given the allele frequencies can be determined by taking the derivative of the expression above with respect to p. Okay, so what I want to do is actually walk us through the calculus here, and to just kind of give us a brief refresher for people that either haven't taken calculus in a long time, or even if you never took it, let's just give a brief refresher about what derivatives are telling us, okay, and why we're going to go through this math. So imagine just some hypothetical function f of x up here in the top panel, and this is the shape of that function, okay. A derivative is then the slope of a tangent line at any point along that function, okay? And what this kind of tells us is the instantaneous rate of change of that function at that point in time. So what I've done here is I've just drawn, it's about 0.42 for this function. I've picked the point at which the slope is equal to 0. And so it's just a flat line, that the tangent line sits right flat on top of that slope. What that's telling us is that the function at that point in time, the rate of change is 0, okay? If we go to the left, then you can see the line is going to start going up like this, and so the rate of change becomes positive. If we go to the right, the rate of change at any point is negative, okay? So again, you can think about the derivative as the instantaneous rate of change at some point in time along that slope. So to connect this back to how does fitness change with allele frequency? You can think about it as at any particular frequency of p, what is the rate of change in mean population fitness? How fast is it changing? How slow is it changing with any given value of p? All right, that's what that tells us. So that's what we're going to derive here so that we can know how quickly is mean population fitness changing as a function of allele frequency. Hopefully you followed that. And let's dive into the calculus here. First thing that we need to do is remind ourselves of something called the power rule. So in calculus, the power rule states that if you have some function that is x raised to an exponent n, then to take the derivative of that function, that is, you move the exponent in front of the x, and then you take 1 away from the exponent. Okay, that's getting you the derivative of that function. So plugging that in to our little equation down here, we're going to first take the derivative of this very first part. Remember, we're just taking the derivative of p. We're not going to take the derivative of w. We'll talk about what that means at the end. So we take the derivative of p. We just move the 2 down in front, and then we take 1 away from the 2, which just leaves 1. And so the derivative of this first part is 2pw11. Piece of cake, super easy, did the derivative of that one. Now, we're going to do a little bit of rearranging in the second part, and we're going to see why. It's going to make some of the math a little bit obvious later on. So we're going to swap where the p is. We're going to move the p to the back, move the w12 to the front. Okay, that's all we've done here, just rearranged a little bit. And then we're going to do the same thing with the last term. We're going to move the w22, or the fitness of the alternate allele. We're going to move that to the front. So now we have the next step. Okay. Now let's get into taking the derivative of these other p's that are left. Okay, so to do that, to do the middle one first, we have to rely on another rule from calculus, and this is the product rule. So the product rule states that if you have two functions, x and z, to take the derivative of those multiplied together as a product, then you take the derivative of the first one multiplied by the second one, plus the first one multiplied by the derivative of the second one. Okay, and that's what we're doing here. So we are taking the derivative of the first part, the 1 minus p here, multiplied by the second one, just p, plus 1 minus p multiplied by the derivative of the second one. So that's just the product rule that we're doing right here, plus the derivative of 1 minus p squared. We've just put the derivative here. We're going to use a different rule for dealing with this derivative. Okay, so before we talk about that one, let's take the derivative of this part here. So the derivative of a constant is 0. So the derivative of 1 is 0. The derivative of a line, which is just p, is 1. All right, so this becomes 0 minus 1 here in the middle. Then we just keep the p, plus 1 minus p multiplied by the derivative of p, which is just 1. So that gives us a solution for this middle part, okay? Next, we're going to use a little fancy rule called the chain rule to deal with this 1 minus p squared. So the chain rule states that if you have a function inside of a function, to break that up, you can take the derivative of the outside function, multiplied by the inside, multiplied by the derivative of the inside. So that's all we've done here. So we're taking this 2, moving it out in front. That's where that is. And then we're keeping 1 minus p, multiplied by the derivative, which is just, again, that's the derivative of the first part, multiplied by the derivative of the function inside of it, 1 minus p. Okay, so we're just using the chain rule to get rid of that 1 minus p squared. So then we are just going to clean up the middle a little bit here. So remember, it's like 0 minus 1 plus 1 minus, so we just cleaned up the middle, and that gives us 2w12, 1 minus 2p. Okay, that's all we've done here is just clean the middle up. So then we take the derivative of this last little part here, which is, since this is a constant, it's 0 minus 1. And then all we're going to do is move that negative to the front, and then we're going to distribute out that 2pw22. And that leaves us with this expression here. Okay, so this is now the derivative has been taken for all of the respective terms. Now we're just going to do some quick little algebraic magic, factor it out, and that gives us with this nice final solution. This solution is in a nice form because on this first term here, we can see that we have the homozygous plus half the heterozygotes minus the second form, which is the other half of the heterozygotes plus the alternate homozygote. Right, so this is a form that's nice to sort of have this in. Okay, so that was a lot of math. So let's kind of remind ourselves once again what we were trying to do. Right, now that we have the derivative of the mean population fitness, we can infer how the rate of mean population fitness changes as a function of allele frequencies. So the shape of this function is a feature of the degree of dominance. So we can think about the additive case in which the heterozygote is half the fitness of the homozygote. This gives us a linear increase in mean population fitness. Okay, so we can see in this top panel here that increase is just linear across time. And in fact, the derivative of a linear function is a constant. Okay, and it's just equal to, in fact, it's just equal to the slope, right? So that's a very simple case in the additive. But if it's not additive, let's say the beneficial allele is recessive, for example, so h is equal to 0, that gives us a different shape. So we see the mean population fitness on the bottom changing as a function of p has a different nonlinear shape, and it increases almost logarithmically, right? So you have this very slow, gradual increase in which the bulk of the change starts happening once the allele reaches appreciable frequency. So with this in mind, we can kind of start to intuit a kind of rigorous definition of what we mean by adaptation, which will become explicit here shortly. So implicit in our model so far is that selection is acting on survivorship. Okay, so that's that zygote to adulthood. That's what we would call viability selection is in that time frame. Okay. Furthermore, our model is presently assuming that genotype fitness is constant and independent of the frequency of each individual genotype, right? And we call this frequency independent selection. So remember when I said before we were taking the derivative of mean population fitness with respect to P, but we were ignoring W. That is to say, we were considering that W is a constant, that it was not going to be changing as the genotype frequency changes. Okay, that's frequency independence. We're going to see that if we relax that assumption, we can get some wonky results, and we'll talk about those when we get there. Furthermore, importantly, since selection acts on genotypes, right, not directly on alleles, assuming Hardy-Weinberg equilibrium, we can still calculate the marginal fitness of each individual allele. Okay, so we can define the marginal fitness of alleles like follows. So whenever you see this little asterisk next to the fitness function, that means we're defining it as marginal fitness. Okay, so W1 asterisk. This is the marginal fitness of allele A1, and that is equal to the probability that this A1 allele pairs with another A1 allele multiplied by the fitness of that genotype, right? Because if you paired with the same one, then you are a homozygous for A1, so your fitness is then W11, plus the probability that you paired with an A2. Okay, so that means the probability that you became, that the offspring is heterozygous, multiplied by the fitness of the heterozygotes. You do the same with W2 two asterisks, so the marginal fitness of the alternate allele. And then assuming random mating, we can actually write the marginal fitnesses as follows. Remember, because under random mating, the probability that you pair with another genotype is simply equal to the frequency of that allele in the population. Right, that's what random mating is telling us, is that probability should be the same. So then we can actually write these fitnesses as follows. Now, since we've defined the fitnesses, the marginal fitness of each one of the alleles, we can then express these as a function of the mean population fitness, right? So the mean population fitness, focusing on the marginal fitnesses of alleles, is then just equal to P times the marginal fitness of P plus 1 minus P times the marginal fitness of 1 minus P. Okay, so can we also think about the rate of change given the marginal fitnesses of these alleles? Well, yes, we can just plug in each one of these marginal fitnesses into the expression that we got previously, do a little simplification, and we find that the rate of change in the mean population fitness relative to the change in frequency is simply equal to 2 multiplied by the marginal fitness of A1 minus the marginal fitness of A2. So now the way that we can interpret this marginal fitness is as the expected number of descendants of A1 into the next generation. So this is a pretty general result that is a very useful way of thinking about fitnesses as the sort of probabilistic statement about how likely any given allele is to leave descendants into the next generation. So in the previous one, we were looking at genotypes, and in this one, thinking about marginal fitnesses, we are looking at the changes in alleles. So why do we care about this? Why do we care about marginal fitnesses if we already have a genotypic fitness? Well, this actually gives us something very, very useful, and it shows us why scaling fitness any way we want doesn't actually impact our results. So I've just listed the two solutions that we found so far for the mean population fitness and the derivative of mean population fitness with respect to P. That's the 2 up here. And so if we take n sub 1 as the actual number of A1 alleles in the next generation, then n sub t is equal to n sub 1 plus n sub 2. Therefore, P sub t is equal to n sub 1 over n sub t. So we can write what the frequency of P should be in the next generation as P sub t plus 1 is equal to n sub 1 multiplied by the marginal fitness of the A1 allele divided by the total number of alleles multiplied by the mean population fitness. Now, since n sub 1 over n sub t is literally the frequency of P sub t, right? That is what that is. The number of A1 alleles divided by the total number of alleles is the frequency. Therefore, we can actually just cancel that out and place a P in its spot, right? So then we end up with P sub t multiplied by the marginal fitness of A1 divided by the mean population fitness. This is why we can scale fitness any way we want, because the ends always cancel out. Okay, so now let's think about how does the allele frequency change from one generation to the next. We often denote this as delta P, where delta often in mathematics just means the change. So delta P is equal to P sub t plus 1 minus P sub t. That's literally just what it is in the next generation minus what it is in the present, is then equal to what we just derived, minus P sub t multiplied by the mean population fitness, which then gives us P multiplied by the marginal fitness of the A1 allele minus the mean population fitness divided by the mean population fitness. This result is very robust, and this particular way of calculating the change in frequency is true irrespective of if the population is in Hardy-Weinberg, whether whether selection is frequency independent or dependent, or if there are many, many alleles. Right, this expression will still work. You will still get the correct change in frequency, expected change in frequency from generation to generation. So this is a really cool sort of central result that we can calculate the change in frequency given that we have some estimate of marginal fitness and mean population fitness. Okay, so all of that was derived assuming only the A1 allele. What we want to do now is actually bring back in the alternate allele, and to do that, we'll just add back in this expression from mean population fitness, and we'll plug it in right here. Okay, so we're just bringing it down, plugging it in here, there it is. We're going to do a little bit of factoring here, and this gives us p1 minus p multiplied by the difference in the marginal fitnesses between the A1 allele and the A2 allele divided by the mean population fitness. Now, this also shows us something pretty cool. Depending on the differences in these fitnesses, we can actually get some pretty interesting shapes. So one of them is that as the frequency of P increases, shown down here on the x-axis, the rate of change of P, shown here on the y-axis, will actually change, right? So, for example, you can have a really rapid rate of increase when P is at low frequency, but once P kind of reaches an intermediate frequency, the rate at which it changes starts to go down, right? And it actually starts to fall. Now, it's still positive, right? So it's still being selected upon and it's still going to go to fixation, but the rate at which it goes to fixation starts to kind of fall off. And we can gain some intuition in why this might be by just thinking about how selection is acting, right? So selection is acting on genotypes. And depending on the degree of dominance, that's going to tell us how selection is going to be able to behave. So we can think about, for example, if the beneficial allele is recessive, right? Then what that means is that at very, very low frequencies of P, most of them are hidden. You don't see them because they're in the heterozygotes. So their frequency increase is very slow initially. But then as they start to get in more and more and more copies, more of those heterozygotes, more of the P's are in the homozygous state. And when they're in the homozygous state, selection can see them. And then they will rapidly increase in frequency. Alternatively, if they are dominant or additive, then what they will tend to do is increase in frequency very rapidly at first, and then they will slowly start to level off. And the reason that they slowly start to level off is that alternate allele, right, the A2 allele that's being selected against, persists because it's hidden in heterozygotes, right? So since most of the time when that A2 allele is present, it's in a heterozygous state, selection can't see it. So the rate of change at which that allele is going to fixation just slows down. And what this often gives us is these kind of sigmoidal curves, right? So the rate starts slow, increases rapidly, and then will start to slow again, right? So again, all of the shapes of the change in P we are getting from the derivations we've just done and from the expressions that we've just shown here. Okay, so long as fitness is independent of frequency, we can combine the two expressions we just derived into one to get a new change in frequency. And by combining them together, we get the change in frequency is equal to P minus 1 minus P divided by 2 times the mean population fitness, multiplied by the derivative of P with respect to mean population fitness. But do a little bit of simplification, and we can see that the change in the frequency of P can be written as P1 minus P divided by 2 multiplied by the derivative of P with respect to the natural log of mean population fitness. This, ladies and gentlemen, is Wright's adaptive landscape. It links population growth, which is that natural log and mean population fitness, right, which is equal to R, with the variance in genotype frequency. So where did that come from? So this P1 minus P over 2, this is actually equal to the variance in the genotype frequencies. So the change in P from one generation to the next is simply the variance in genotype frequencies at that particular time point multiplied by some fitness function. And that fitness function is represented by this derivation here. So this enables us to envision changes in allele frequencies as a function of a fitness landscape, okay? And that fitness landscape, the shape of that fitness landscape is dictated by this function, right? So whatever values we plug into this function is going to change the shape of that landscape. Furthermore, what this tells us, what Wright's adaptive landscape tells us, is that populations should be climbing to the local maxima of mean population fitness. Okay, so long as this expression holds true and fitness is independent of frequency, populations will climb these little hills until they reach the top, and that top is represented as the maxima of mean population fitness. Hence, we can rigorously define the concept of adaptation as the evolution of traits that permit organisms to maximize the population growth rate in a given environment. So we see that ideas like adaptation and Wright's adaptive landscape have theoretical, mathematical underpinnings that allow us to investigate how they should change in response to some fitness function, right? And that fitness function, again, defines where that population should end up with respect to genotypic variance. So with that in mind, let's think about the different kinds of fitness functions and think about them in respect to Wright's adaptive landscape metaphor. Some features of these adaptive landscapes are actually unstable. And this means that the direction of the change in frequency depends on the starting condition of P. To grok what I mean here is imagine you have a population and there's a lot of variation segregating neutrally in that population, and then that population enters a new environment. And now suddenly selection is acting on one of those variants, right? So the frequency at which that variant is at when they enter the new population, depending on the fitness function, can dictate what's going to happen in the population and where the local maxima of that fitness landscape they land on. So the simplest case is in directional selection, okay, where there's only this one very obvious peak, and it peaks when the frequency of P is 1, all right? That means that directional selection should always move a population to maximum fitness, and there is always an equilibrium that exists, and it's irrespective of where you started. Okay, so no matter if you start at very, very low frequency or very, very high frequency, you're going to go to fixation. So that's represented up here. So you can imagine the little red ball is the starting frequency of P. X-axis here is P, y-axis is just the mean population fitness, and you can see that it should always increase, right? And it's going to eventually increase and reach the maximum either when P is fixed or when P is at very, very high frequency. So that's the peak of that landscape. Alternatively, you can imagine stabilizing selection. So stabilizing selection favors alleles that are at intermediate frequency. And again, irrespective of where we start, they're going to move to the same mountain. So if we start, for example, our little red ball, if we start at the frequency here, selection is still moving you up, and so you're going to land in this top spot. Now, this one's pretty interesting because when you reach that peak, right, any additional increase in your frequency actually decreases mean population fitness, right? So it decreases it, and so what that's going to do is now selection is going to push back, and it's going to reduce your frequency, right? And so what you end up happening is you end up just stabilizing on the top of that hill, okay? So that's stabilizing selection, is stabilizing those frequencies at the top of the hill. Alternatively, disruptive selection or underdominance, this is where selection is acting against alleles at intermediate frequency. The equilibrium that is reached is entirely dependent on where we start, okay? So imagine we started P right here. Any increase in the frequency of P decreases mean population fitness, right? And so what P is going to need to do is decrease in frequency. And so the equilibrium for this particular function, if we start here, is right here, right? It's going to move that allele to up here. But what if we started P on the other side? Now, any decrease in the frequency is going to decrease mean population fitness. And so the equilibrium is actually on this peak, right? Similarly, if we start down here, any increase is a decrease in mean fitness, so it's going to push it downwards. So what this shows us is depending on where we start, right, what that allele frequency is at when we begin our iteration or when selection is acting, is going to dictate where the equilibrium in that population is. And in mathematics, anytime that you have an equilibrium that is always reached, it's always the same one, irrespective of where you start, we call that a stable equilibrium, right? So whether P is 0 or 1, it's always going to reach the same equilibrium frequency that has the highest mean population fitness. Alternatively, in the case of disruptive selection, it is an unstable equilibrium because the initial conditions are going to dictate where you end up in that fitness landscape. So again, we have two potential landscape topographies that lead to stable equilibria, and then one that leads to unstable equilibria because it depends on what the frequency of that allele was when we started. Okay, we've been talking thus far about frequency independent selection, where the fitnesses of each one of the individual genotypes are considered to be independent of that genotypic frequency itself. However, in nature, this is often not the case. Much more likely is that there's frequency-dependent selection. That is, the fitness of each one of those genotypes changes as a function of the genotypic frequency itself. As just sort of a simple case, this is often applied in ecology. So you can imagine a population that is has two different kinds of, let's say, feeding strategies. Some that will share and then some that steal, right? When the sharers are at very, very high frequency, then the stealers have the highest fitness, right? So they can rapidly increase in frequency because there's lots of sharers that they can steal food from. But as their frequency gets too high, they start to lose the sharers, and now being a stealer is not as good, right? And so their fitness starts to fall off, and then the fitness of the sharers start to increase. Right. So that's just kind of a simple ecological case. It's frequency-dependent selection is used widely in game theory, behavioral ecology. A lot of fields think about frequency-dependent selection. So that's just one kind of simple example. So in this case, if we go back to when we took the derivative of the mean population fitness, that's what's shown up here at the top. Now all that we need to do is then take the subsequent derivative of the fitnesses themselves. Okay. So instead of just taking the derivative with respect of P with respect to W, we also need to take the derivative of W. And so that's what's shown here. It's just us walking through each one of these and taking the derivative. The important thing to bear in mind here is that in the case of frequency independence, that means that these values are constants, right? And if these values are constants, this term becomes 0. And it just cancels out, right? And then you're just left with up here. So I just wanted to kind of show you that frequency dependence only emerges when when the W11 actually does change with respect to P. So there's some shape function within that fitness value. So what we want to do here, just for kind of simplicity's sake, is actually just take the sum of all of these values. So this is the average of the derivative of genotypic frequencies with respect to allele frequency, and we will just rewrite that as the expectation of the derivative of genotypic fitness, which is W, with respect to allele frequency, which is P. So this is the term we're going to substitute this blue box with. And just remember, when you see this term, this is actually what it is, right? It's the summation across each one of these terms, okay? So now we can then take our little variable that we just created, which again is a summation of these terms, and we can plug it in to Wright's adaptive landscape. And when we do that, this is what we end up with. So this is Wright's adaptive landscape on this part, and then we're just adding in, or in this case, subtracting from the contribution to frequency dependence to the rest of the change in allele frequency. So taking a second just to kind of remind us what all of these terms are. So in the red box, this is the additive genetic variance of fitness. Okay, so remember I told you before that P1 minus P divided by 2 is the genotypic variance. And so all that we've simply done is we've multiplied the genotypic variance by 1 over the mean population fitness. When you multiply those two together, you get P1 minus P over 2 times the mean population fitness. Okay, so that's what that term is. That's the additive genetic variance of fitness multiplied by the average change in genotype fitness with respect to the change in allele frequency. That's what this second term here represents. Okay, so just to put it into words so that we can remind us what all of these values are actually representing. So let's think about a couple of different shapes that this fitness function could take, and then see if we can intuit what might be a stable versus an unstable equilibrium in this setting. So first is just a very simple directional selection case where the fitness function is a constant, and irrespective of where we start P off, that is whatever the frequency of P, doesn't matter, it will increase linearly. And that's what's shown here. So we're just going to increase, and then the stable equilibrium point is the fixation of P. That's directional selection, very simple. Now let's imagine the slightly more complicated case of negative frequency dependent selection, where the fitness of P is at the highest when P is at low frequency, and then as P increases in frequency, eventually its fitness levels and then will go down, okay? So if we started here, for example, P is at high fitness, so it's going to increase in frequency. But as it increases in frequency, it eventually reaches a point to where any further increase in frequency actually decreases its fitness. And so what that means is that it's going to stabilize here. Similarly, any decrease in frequency increases its fitness, so they have more offspring, which increases its frequency, right? So what you end up with is this kind of stable equilibrium point at which it's not going to go any further. It's not going to increase beyond because that decreases its fitness. And it can't go backwards because as the frequency goes down, its fitness goes up. And its fitness going up means it's increasing in its frequency, right? And so it gets stuck in this stable equilibrium right here where it can no longer be at higher frequency. This is an important point and something that only emerges in frequency-dependent selection. Notice that the highest fitness point is actually back here. This is where the fitness is the highest. But notice that this is not a stable equilibrium point and that P will not stay there. Instead, it's going to increase in frequency until you get a stable fitness function that is not the maximum of the possible mean population fitness. Hence, in this case, in the case of frequency-dependent selection, selection is not guaranteed to maximize population fitness like it did in the frequency-independent case. So now let's look at the case of positive frequency dependence. Again, we can see here that there's a strong dependence on where we begin, what's the initial frequency of P as to where we're going to end up. So if we start off on this side of this unstable equilibrium, right? So if P is over here, P's fitness is low and it's going to decrease, right? And eventually it's going to decrease and the stable equilibrium is loss. If we start off over here, right? At high frequency, then its fitness is high and it's going to increase and it's going to stabilize at fixation. Okay. The only equilibrium point that can be achieved in positive frequency dependence is if the alleles begin at intermediate frequency. That is, if this is the starting condition of the population. So while those are obviously more complicated than the frequency independent case, it can get even more complicated. You can imagine any kind of fitness landscape, any kind of fitness function could describe this system. So here's an example of just a complex frequency dependence where the fitness is actually sort of oscillatory across different frequencies, right? And so here, the equilibrium frequency is strongly dependent on where we begin, right? So let's say we began at high frequency. Right? At high frequency, we are at low fitness, and so we're going to start to go down. Okay, we're going to start to go down, and then we're going to reach sort of a stable equilibrium point right here. Because the stable equilibrium point is achieved because any further decrease in frequency increases fitness, which pushes us back to equilibrium. Okay. Similarly, if we were to start right here in the middle, since we're at the highest fitness, we're going to be pushed forward, and then we're going to stabilize right here at this equilibrium point. Because again, any additional increase in frequency decreases our fitness, which pushes us back to the equilibrium point. If we, however, start here, right, now we are at low fitness, and so selection is going to push us down until we eventually reach this stable equilibrium. Again, because any additional decrease in frequency increases fitness, which pushes the frequency back down. Okay, so we can see there's multiple stable equilibrium points as well as multiple unstable equilibrium points. So this is, in many ways, a kind of counterintuitive result, right? That Darwinian selection, this adaptive process, can actually drive populations to not sit on fitness peaks, right? And they can actually get stuck in these kind of fitness valleys. And indeed, not only can populations get stuck in fitness valleys, they can even be driven to extinction by positive natural selection. We call this process Darwinian extinction, and the traits that drive it we call kamikaze traits. And again, this kind of thing only happens in frequency-dependent selection, but it is a pretty wild phenomenon. And it's akin to a kind of Fisherian runaway selection. So imagine, for example, this is just a very simple example. Imagine you have a population of weevils that eat leaves, okay? And most of the individuals in the population eat leaves of a very specific plant, okay? Now, some of the individuals of the population gain an adaptation that permits them to eat seeds of the plant, okay? So initially, they are going to have very high fitness because they have this completely new food source, right? So if we think about red as the fitness of the seed eaters and then green as the fitness of the plant that they are eating, right? So initially, the seed eaters have very high fitness, right, at very low frequency, which stays pretty high. But eventually what ends up happening is they're eating the seeds of the plant that needs to reproduce. And so the plants are now not reproducing as much because they're losing their seeds that are going to contribute to their reproduction, right? So the population of the plant starts to go down. The beetles continue to persist as that population gets lower and lower and lower and lower, until eventually that population gets so near to extinction that the beetles themselves can't persist and will drive themselves to extinction as well. And so this way is just sort of a simple example in which how a predator can drive its prey to extinction, and in doing so drives itself to extinction, right? So this is, again, is a case of Darwinian extinction that's driven by these coevolutionary interactions between these two organisms that lead some individuals to do better, but in doing better, they are actually destroying the environment that they need to live in. Cancer is another sort of empirical example of natural selection actually leading to its own death, right? Because cancer, the environment in which cells live, you can think of as basically the individual, right? And so as they spread across that environment, they will eventually kill the environment that they need to live in. And that is a case of positive Darwinian selection. They're evolving under natural selection, and yet they're destroying the environment they live in. So again, this is a case of frequency-dependent selection being capable of driving not only adaptation, but also extinction. Now what I want to do is just sort of bring everything together that we've learned so far, all the things that we've derived, all the concepts that we've introduced, to help us understand one of the most important ideas in early population genetics. And this is Fisher's fundamental theorem of natural selection. So to understand what it is, what it means, let's begin with the derivation we just did for frequency-dependent selection in Wright's adaptive landscape. So again, that's this here. In words, this is the change in P given the variance in genotypes, which again is this, and frequency-dependent selection, which is this term. The next thing that we need to think about is that we are often thinking about fitness as a mean, which we can call W. And since it is a mean, that means that that fitness effect has a variance, okay? And we call the variance in genotypic fitness the additive genetic variance of fitness. Now, recalling that this value in blue is the variance in genotype frequencies multiplied by 1 over the mean population fitness, we can actually rewrite this change in the frequency of P with respect to the slope of a regression line of fitness against genotype. So I'm showing that here. So this is A2A2, A1A2, and A1A1. This is the black dots represents the estimated fitness for each one of them. So that's there in W. And then we can draw a regression line between them, right? And this regression line is determined by a least squares approach. And you can see that there's some distance between the observed and the line that represents the least squared, or the best fit line, okay? And this best fit line is determined by a slope that we're going to call beta WG. And so what this beta WG represents, what this slope represents, is actually this function, right? It's the rate of change that we expect to see given the genotypic variance and the mean population fitness. So this is one kind of way that we can actually get at this fitness function, is by estimating it via linear regression. And so we can write this again as the change in P is 1 over the mean population fitness multiplied by the variance in genotype frequency, which again, that's just this term here, multiplied by this beta coefficient, right? Which again is just this here. And we can rewrite this as P1 minus P over 2 W bar multiplied by the beta coefficient. So putting all these equations together, we can show that the variance in genotypic fitness is equal to the mean population fitness multiplied by delta P, which is the change in the frequency of P, multiplied by that beta coefficient of that regression line. And that, again, that beta coefficient simply represents the fitness function, which is right up here. Right, this is what we derived previously, where this part of the fitness function represents frequency independence, and the second part represents frequency dependence, okay? So we just plug that in for the beta coefficient. That gives us W bar times delta P times that fitness function. Now we can do a little simplification, and if we recognize that delta P is usually pretty small. And if that assumption holds, then delta P multiplied by this first derivative is approximately equal to delta W bar, or the mean change, or the change in mean fitness. Furthermore, we can rewrite delta P multiplied by the frequency dependence as simply the expectation of delta W. Then we can rewrite this whole little function here as the variance in genotypic fitness equal to mean population fitness, which is there, multiplied by delta mean population fitness, which was this assumption we made here, minus mean population fitness multiplied by the frequency dependent component. Now, if we are interested in the rate at which mean population fitness should change from one generation to the next, we just rewrite this and solve for delta mean population fitness, or delta W bar. And in doing so, we see that delta W bar is equal to the variance in genotypic fitness divided by mean population fitness plus the expectation of delta W. And this, ladies and gentlemen, is Fisher's fundamental theorem of natural selection. Put in words, it is the change in the mean population fitness equals the genetic variance in fitness. Very often in textbooks and papers, you will actually see them drop the second term. The frequency dependent term just won't exist. And so all that they will show is this first term here. And so if we discard that term, Fisher often called this second term actually the environmental deterioration. So you can think about the move away from the fitness optimum due to some environmental effects, right? But if we ignore that term for a second, as Fisher did and as a lot of other population geneticists do, and we just focus on this part, then we can kind of understand why Fisher thought it was so important. One is that since variance can never be negative, and ignoring frequency dependence, this means the change in mean population fitness is either stable, it's not changing, or it's increasing. That is to say that if there is any variance in a population for fitness, right? If there's any variance in fitness, then the mean population will increase in fitness. That's what that's saying. There's variance in fitness, natural selection leads to an increase in the fitness of the population. Okay, that's the very simple assumption that's being made. If there's no variance in fitness, then there's no positive selection. Selection is not climbing any optimum. Now, this could be either because it's already at an optimum, right? So it's not that there's no selection at all. It could be that it's just sitting at an optimum already, or there is no variance to allow it to reach some optimum. So again, Fisher thought that this was, Fisher called this literally the fundamental theorem. He thought this was akin to thermodynamics, and that it was sort of a fundamental principle in population genetics. And again, this fundamental principle is the idea that natural selection should act to maximize population fitness, and that its ability to do so is directly related to how much variance in fitness there is at any one time. Okay, that's cool, but one of the things that we need to remember, of course, as we've already talked about, is this second term, right? This frequency dependent term. Very often, people will shit on the fundamental theorem and say, oh, it's not really that fundamental, because, for example, in populations, fitness can go down. And that could be, and to them, that sort of undermines the concept of the fundamental theorem, at least in the way that Fisher kind of advertised it. And this does not, decreasing fitness does not contradict the fundamental theorem, because the fundamental theorem actually does include this second term, okay? And the second term accounts for the fact that if there is frequency dependence, you can actually get a decrease in mean population fitness, okay? So while that term is often ignored and thrown aside and not considered, technically it is not a violation of Fisher's fundamental theorem if mean population fitness goes down, because there could just be frequency dependent selection going on. And we've already showed that in the case of frequency dependent selection, it is not guaranteed that there will be a maximum fitness attained, right? That the local maxima of mean population fitness is not guaranteed to ever be reached in the case of frequency dependent selection. So I don't think that undermines the fundamental theorem. The universality of the fundamental theorem is a product of, or is a function of, how many of these assumptions that we are willing to accept. So that's one of the things that, it's a useful expression. It's a useful way to kind of maybe think about evolution, right? That natural selection depends on there being some variation to act upon, right? But in terms of like how fundamental is it, I leave that for the audience to decide. Okay, so we talked about a ton of things here. We've gone through a lot of math. A lot of it was probably really, really dense. Hopefully you've picked up something. And I want to use this as kind of to just sort of bring everything together, refresh our memories on all the things we've talked about so far. So first, we began with sort of defining fitness. And we showed that fitness can be defined in terms of the per capita population growth rate. And that is that we can directly link genotypic fitness, that W bar, we can directly link it to ecological principles of population growth rates. Population growth rates are often denoted by R, and that is approximately equal to the natural log of mean population genotypic fitness. So fitness is not some arbitrary thing. It does have an ecological meaning that is of relevance and thinking about the rate at which populations increase or decrease in size, etc. Secondly, we get kind of a rigorous definition of adaptation as a process by which the population growth rate increases and is maximized, reaches a maximum as a function of increasing allele frequencies, right? So again, Wright's adaptive landscape showed that population should be maximizing fitness. They should be searching out the peaks in that landscape, reaching that maximum fitness, and in climbing that hill. That is the process of adaptation. That's what we mean by adaptation, is the ability of increasing allele frequencies to maximize fitness. Again, we derived Wright's adaptive landscape, and then we talked about how this second term in it, this derivative term, is the fitness function that dictates the shape of the fitness landscape. And then the first term is just the variance in genotypic frequencies. Then we showed how frequency-dependent selection can actually lead to unstable equilibrium in which fitness maximization is not guaranteed, and that allele frequencies can actually fluctuate perpetually. So allele frequencies can fluctuate perpetually, or they can reach sort of a stable equilibrium that is some value below the maximum population fitness, okay? So there are peaks elsewhere that in the case of frequency dependence are inaccessible by natural selection. We derived Fisher's fundamental theorem, and we showed that it states that the mean population fitness is always an increasing function of the additive genetic variance in fitness. We also showed that there's some caveats there, right? That the fundamental theorem also includes this frequency dependent component, and hence it's not necessarily a violation of the fundamental theorem for fitness to be decreasing, or for a population to not be able to achieve that maximum fitness because of frequency dependence, right? So we sort of gave that caveat. And so here at the very last point that I want to make is to try to dispel these sort of simplistic notions of natural selection. Obviously, this is a pretty dense video where we went through a lot of math and a lot of like difficult topics and terminologies, etc. But the purpose here is to show that the concept of like survival of the fittest, right, is a gross oversimplification of natural selection, and that in fact it's such a gross misrepresentation as to be wrong in many, many cases. That we actually have no mathematical expectation that natural selection should necessarily maximize fitness, right? And we've shown that in many different ways in this video today, that while the tendency of natural selection is to increase mean population fitness, there's actually no expectation that it should maximize it, okay? Especially in the case of frequency-dependent selection. A couple of other sort of caveats that I want to put on this is that in this video, we've only looked at one locus models, okay? And that's true of the previous one as well, but I want to point out that natural selection as a concept can also be applied to two locus, genome-wide, multiple genes. So like we did the very simple kind of base, laying the groundwork for some concepts that we will talk about later, especially thinking about how natural selection interacts with some of these other mechanisms, right? So in this case, natural selection was the only force acting. But you can imagine when you start to add in other forces, things get even more complicated, right? And that's quite frankly, that's when it really gets exciting. That's when the videos are going to really get fun, is when we start putting in all these mechanisms together and see what happens, right? So I want us to bear in mind that that's coming, and it's going to be a bit more complicated, but I think it'll be a useful sort of exercise. Yeah, so that's the next mechanism of evolution, and that is evolution via natural selection. Like I said, this is a pretty dense video, so if you have any questions, please feel free to drop them in the comments. I do want to shout out Sean Price's Evolutionary Theory book. This is where a lot of this has come from. I got this book relatively recently. This is an excellent book for diving into the mathematical ideas behind evolutionary theory. So if you're really interested in this idea, in this concept, I highly recommend this book. And that's where a lot of sort of the derivations here come from. So yeah, thanks so much for being here. I hope you enjoyed it, and I will catch you next time.